Today we start the inherited change video number 4, and we will continue with the dihybrid cross and one other example of that, and then we will go on to the next topics of this chapter. One of the different examples for a dihybrid cross is when we cross. Now if you look at it, and I would in the question it would be heterozygous for both characteristics. So it is heterozygous for the purple stem. This has been continued from the previous video. So it is heterozygous for purple stem, and it is also heterozygous for the cut leaves. So these were the dominant traits, purple stem and cut leaves. And then you cross it with a homozygous recessive. So homozygous recessive means this would have been a green stem, and this one would have been potato leaves. Now when you cross this, it's very obvious that the ratio here is going to be 1:1, ratio 1, ratio 1. Why? Because it's very this one would always give 4 types of possible gametes. So I always number these and I say 1, 2, 3, 4. So the first one is going to be 1, 3, then 1, 4, so big A, small d, and then 1, 3, 1, 4, then 2, 3, and then 2, 4. So we have big A, big D, then big A, small d, so big A this one and big D, then big A, small d, then small a, big D, so it would be this one, and then small a, small d. So what we have to understand is that we have to make the 4 possible gametes. So in this situation we have big A, small a, big D, small d. So the first one is 1, 3. Please remember this for whenever you're doing this. Then 1, 4. Then 2, 3, and then 2, 4. So if we give these numbers, this one is 1, this one is 2, this one is 3, and this one is 4. So these are the 4 possible gametes. But in this situation, in which we have this here, there is only one possible gamete because everything else is the same. So the only possible gamete is this. Now you have to remember is that you have to circle the gametes. That's how we usually write it. So we circle the gamete. So the 4 gametes for this would be this, and the 1 gamete for this would be small a and small d, and you circle this, or you circle it wherever you're going to write it. Now as we look at this, this a complete table is that we crossed a known which is heterozygous for both characteristics with one which was homozygous recessive for both characteristics. So the 4 possible gametes for this were 1, 2, 3, 4, and then because this one only had 1, so I've written this here, this one has been written here. So we make, you can write this 4 times, it comes to the same thing. It will be 4:4:4, or 1:1:1:1:1. So now you can see how I have written all these. So this one, the first one, this one would be heterozygous for both characteristics. Now this one is heterozygous, one is homozygous recessive. Then this one is homozygous recessive, this is heterozygous, and this is homozygous recessive for both characteristics. So we get a ratio of 1:1:1:1 whenever it is, number 1, heterozygous for both characteristics crossed with a homozygous recessive for both characteristics. So please just remember that, pause it and just make your notes, and then you will, of course, it will be easy for you in the exams to do questions on genetics. Then we start discussions on sex-linked diseases. Now first of all, we've got to be very clear, in every human cell there are, as you all know, 23 pairs of chromosomes. Well, that's fine. We have 23 pairs of chromosomes in every cell of our body. Well, that is probably, we teach it to you at O levels. But the interesting part is that it is slightly different in a male and slightly different in a female. And the male cells have got less DNA as compared to a female cell. And the reason for that is very simple. Male cells have 22 pairs, yes. There is a 23rd pair also, but that is the one which decides the gender. So in women also, in the female also, there are 22 pairs. So the 22 pairs in the male and the female are the same, and these have been called autosomes. They code for the body characteristics, so that's why they are called autosomes. But then there is one pair which decides the gender. So the male, that 23rd pair, one is a little long and one is a little short. Now we as biologists call the longer one, we've called it X, and the shorter one we've called it Y. But in the female, surprisingly, both are the same length. So if they're both the same length, well, we've decided to call them XX. Now please remember, this is humans. We are talking of humans. In certain animals, it's the reverse. I mean, the XY would be in the female and the XX would be in the male. So it's not for every organism. So in humans, the 22 pairs are the body chromosomes, and those are called the autosomes. But the 23rd pair which decides the gender is also called the 23rd pair of the sex chromosome. So in the male, one is the same length as, you see, so the XX in the female are the same length, and then there's one X which is the same length as in the female, and then there's a shorter one. And the shorter one is called a Y, and the longer one is called the X. And similarly, there are two X's in the female cell. So, I mean, if you do a blood test, you can tell, if you look at the chromosomes in one of the white blood cells. Why we don't look at the red blood cells? Because the red blood cell has no nucleus. So, but when you look at a white blood cell, we will then break open the nuclear membrane and we'll take out the chromosomes and we'll see them, and we find if the 23rd pair is not the same length, one is longer and one is shorter, then we would of course realize that this is a male. These blood cells are from a male and not from a female. Now we need to understand is that there are some diseases which are present on this pair of chromosomes, which is the sex-linked chromosome. So if there is the part of the X chromosome, and then this is the Y one, the Y is shorter. So now the diseases which are present here, the genes which are present here, and if they cause a disease, then that disease would be called a sex-linked disease. So please understand X and Y. But whenever we have to represent these, we will say, like for instance in this disease, which is a disease called hemophilia, and there is a clotting factor which is not produced. The disease hemophilia is a recessive disease. Now this is of course something which I would give you in the question, and this would not be something you need to remember. So this is a recessive disease. Now this of course information would be given in the question. So now you can see we can have a normal female, but the normal female means it has to be a capital H. So the normal female would be X bada H, X bada H. So it's a capital letter. Now the woman who's a carrier would have the X big H and an X small h. But the woman, of course, this woman does not exist on the face of this earth. Why? Because this is a lethal mutation and this zygote does not develop. So that means it doesn't develop into a human being. So there's no such female who is X small h, X small h. Now when we look at the males, you see there's a part of the X chromosome where there is no corresponding part of the Y. So if there's anything written on the X, we just write it X raised to the power H, which means it will be somewhere here. And then there's nothing on the Y. So XH and then nothing on the Y. So the Y doesn't have anything on it. This is the Y chromosome. And a hemophiliac man would have the small h and of course nothing on the Y. So please understand this. Women can be normal, can be a carrier. There's no such woman who's a hemophiliac. And a man can be normal or he can be a hemophiliac, which means he is going to carry the recessive allele. He is going to carry the recessive allele. Now this is a very favorite question which comes on this part of the syllabus is, can a man, so this would be the question which would come many times in your papers, can a man with hemophilia? So if the man is suffering from hemophilia, so that means we have an X and we have a small h and there's a Y. Can a man with hemophilia pass on the disease to his son? I mean, think about it for a while. Just pause the video. Now you realize if you're a son, you got the Y from your dad. So the son always gets the Y from his dad. Now this of course makes it very clear that the Y is going to go to the son. So the males have the Y chromosome, so they are not sufferers. But neither are the female sufferers. I mean, this female is a carrier. This female is a carrier too. So this man with hemophilia is this man. He cannot pass it on to his son because a son is a son because he gets the Y from his dad. But he is of course given it to his daughters. Now these daughters are going to give it to their offsprings. So you can see how this is going to be transferred to the next generation, but just because the grandparent had it. So yes, it cannot be passed to the son, but it can be passed to the grandson. Now this is the person who had the hemophilia. He's passed it on to his daughter. This is the daughter. This is the father. This is the daughter. And now she passes it on to her son. So the question which I asked you, can a man with hemophilia pass on the disease to his son or his grandson? Now this was the explanation for it. Yes. The man with hemophilia, which is this one, passes it on to his daughter, and then the daughter passes it on to her son. So that's why he can pass it on. He cannot pass it on to his son. His son cannot be a sufferer because he gets the Y from his dad. Now I'm going to explain to you how to do genetics questions. Now I'm doing it with an example of blood groups. Now as we all know, there are 4 blood groups. Now the people with blood group A. Now another thing which we've got to remember is that there are 3 alleles. Alleles means the alternative form. So there's IA, there is IB, and I raised to the power O. Now the reason why we're using 2 letters, because this shows a phenomenon of codominance. Because the people with blood group AB are showing both the dominant alleles, so that is why this will be called codominance. So people with blood group A could be IAIA or they could be IAIO. People with blood group B again have 2 options. They can be either IB or they can be IBIO. People with O group have only one option. They can only be IOIO. And the people with blood group AB will be IAIB. So please understand that we have 4 blood groups. So we have 4 phenotypes, but we have 6 possible genotypes. This is one genotype, second genotype, third genotype, fourth, fifth, and 6th, and I always said is you use letters to show genotypes. Now what we're going to do, a genetics question. The question was, how can a couple, what will be the blood groups of the couple if they have 4 children and the 4 of them have different blood groups? The 4 of them have different blood groups. Now you've got to figure out how is that possible that a couple can have 4 children and 4 of them have different blood groups. Now how are we going to do this question? I'm just going to explain it to you. Please remember, genetics questions are marked per line. They are marked per line. So if you've got the entire line correct only, then you get a mark for it. So let's look at it. The first heading, you might write or you might not write the heading, but you've got to remember is that you have to work according to the heading. So the first heading is parental phenotype. Then the second heading is parental genotype. Then the third line is gametes. And the fourth line is offspring genotype. And the fifth line, so we've got one line, second, third, fourth, and the fifth line is offspring phenotype. So please remember, you've got to always answer these questions according to these 5 headings. You might not write the heading. The heading is immaterial if your line is not correct. So if I said a couple has 4 different blood groups, so the possibility of that is if the parental phenotype is if there's one parent A and one parent B. And this is blood group A. So this is blood group A. This would also be blood group A. Right? Now what will be the genotypes? The genotypes will be I, A and IO. Similarly here it will be IB and IO. That's the only way it can be possible. Of course there can be IA as well and IB as well, but then all the children will be IB. Now the gametes, how do we have to write them? We just have to write them separately, whatever you've written in the previous line. And the reason why we're saying that meiosis would result in the gametes having one of each pair. So if this is a male, then the sperms would be of 2 types. Either they have the IA or the IO. Now in this case, if this is a female, the gametes, the ovum would either contain an IB or an IO. Now if you number these again like I did the previously 1, 2, 3, 4, then we have to do it in a very methodical manner. We have to do 1, 3, 1, 4, 2, 3 and 2, 4. So if we join this 1, 3, now you can see how this is going to change. Now if it is 1, 3, then we have IA, the sperm has IA and the ovum has IB, so the zygote will have IA, IB. Now the blood group will be AB. So you see, this was one line, this was the second line, this is the third line, and now this would be the fourth line, and then we've got the fifth line, the last one. And if I continue doing that, so I said 1, 3, then I said 1, 4. This will be IO. This person would be blood group O. Right? Now we have, what did I say? 2, 3, 2, 4. So if we have 2, 3, then it will be this one. And 2, 3 will be IBIO. We always write the dominant one first and then. Now IBIO, there is no such group as BO, so the blood group is B because O is recessive. And then the last one which I said 2, 4, this would be IO. And this would be blood group O. So you see, in this question what I wanted you to understand was that what are the 5 lines in doing genetics questions. When you're doing genetics questions, you've got to be marked per line. Sometimes of course the information is given in the question. So if the information is given in the question, maybe this question could be for 4 marks because this information is given in the question. So there will be a mark for this line, 2, 3 and the 4th line. Now this shows that this couple has a 25% chance of having either of these blood groups. But I mean, if this couple only has 4 children, then that's a very small number. and that is not probably going to apply to it. And I mean, this couple can have 4 children, all of them having blood group AB. Why? Because in small numbers this probability does not hold true. But if this couple has 100 children, that's a bit of a joke, then roughly they would be, the 4 groups would be in a percentage of 25% probability. So there's 25% chance of having AB, there's 25% chance of having blood group O or blood group B or blood group, okay, wait a minute, I've got, sorry, we need to make a little correction in that. You see, this would have been AB, and this was of course 1, 3, 1, this is 1 and this is 3, and then we have 1 and 4, so this would be blood group A and O, and this would be blood group A, so we need a little correction. I think I made a little mistake in the previous one. And then we have blood group B, which is of course right, and then we have blood group O. So 4 possibilities and a 25% probability of having any one of these blood groups. So this is what you need to be understanding about the questions on genetics. The last topic that we handle here is a chi-square test. Now, as you can look at the formula for the chi-square, please remember you're not going to memorize it because this will be given in the question. So there's something known as O, and that O is the observed number. And then there's something E, and that E is the expected number. So O is the observed and E is the expected number. So these are the 2 words that you have to understand. Now it's O minus E whole square, and you divide that by E. First you do, for each one you do that, and you get these values. I'm going to just explain it to you how you get these values. And then you add all of them. So there is this sign of summation, which means you have to add all of these. And this is the sign that is here, that you can see it here. So O minus sum of O is equal to some value, and that value is, in this situation, it is of course is equal to 0.79. So you've added all these. So this was the observed number 86, and then 81 was the expected. So 86 minus 81 is 5, and then square of 5 is, because O minus E whole square is 25. Here you had 26, then 27, then minus 1, 1. Then here you had 24, 27, then 3, and so we got all these different figures. Now, as you look at all these figures, first we have to understand how did we get these observed and expected values. So we crossed two heterozygous. We took a purple cut and we crossed it with another purple cut. The same example that we talked about in the dihybrid cross. Now, if you remember, whenever we cross heterozygous for both characteristics, the ratio is 9, ratio 3, ratio 3, ratio 1. That is the rule. So we actually got 144 offsprings, and then when we planted the seed, so we first crossed, we put the pollen on the stigma, we waited for fertilization and fruit formation, and we collected 144 seeds, and then we planted them. Now these values you cannot figure out because these would be the observed values. So these are the observed values, what we observed. That means we planted 144 seeds in the ground and we let them germinate and then grow, and then we figured out, okay, the purple cut were 86. So this you will never have to calculate, you will always be given this in the question. The observed values will always be given in the question. So this is what we need to understand. So the observed values were 86, 26, 24, 28. Now we needed to calculate the expected values. Now the expected values have been calculated like this. Very simple. Why? Because this is 9. Total 9 plus 3 plus 3 plus 1 is 16. So 9 out of 16 into 144. Why? Because 144 was the total number of seeds. So we got this value of 81. Similarly, 3 out of 16. Why? Because this is the 3. Then another 3 out of 16 and then 1 out of 16. So we figured these out, and these of course is all the calculations is the expected. These are all the expected values because we figured out they were 144. So 9 over 16 into 144, 3 over 16 into 144, again 3 over 16 into 140. Of course you can do it once only, doesn't have to do it twice. And 1 over 16 into 144. So these of course are all the expected values, the E. These are all of course the O values. So now you can understand what have I done in this question. So here I had O 86, right? Then 81. Then 86 minus 81 is 5. And then you square it, so 5 5s are 25. Similarly, we did it on this side and we got 0.04. And then what we did do is we wrote all of these again. So 0.31, 0.04 and 0.33 and 0.11, and you got a value of 0.79. This of course was the answer. This was what is the chi-square value. But what do we have to do with this value? That's of course the most interesting part of this question. Now our calculation of x square was 0.79. Now what do we do with this value? That's the most important thing. What we do is we look at a chart. This will be of course given to you again in the paper. Now there is 1 degree of freedom. Now how do we calculate the degree of freedom? What do you mean I calculate? Means we had 4 values here. 1, 2, 3, 4. So you have to calculate n minus 1. Now here n is equal to 4. So 4 minus 1 is equal to 3. So the degree of freedom will be 3. Once again, n minus 1. Here it was 4. 4 minus 1 is equal to 3. So the degree of freedom will be 3. So we have to look in the 3 lines. So we look at this. This is always different. So if it was 5, then 5 minus 1, 4. If there were 2, 2 minus 1, 1. So the degree of freedom is calculated by first finding out the number of observations they had and then minus 1. In this case it is 3. In this case it is 3. It could be different. It could be 2, it could be 1, it could be 4. Now important is that the probability that we have to consider, it says here probability greater than. Now you see here there's 0.1, then there's 0.05, then there's 0.01 and then there's 0.01. So it is decreasing in this side towards the right. Now what you have to see is you've got to see in this line, but you have to always see in biology it is on 5%. So 0.05 is 5%. Now in 5% the value is 7.82. So now this 7.82 is the critical value. This is called the critical value. Now if your value is less than this, now your value is definitely less than this, so it is probably this side. This side means it is more than 0.1. So it is probably more than 0.1, right? So if your value is less, 7.82 is the critical value, but your value is 0.79, so it is less. So you accept the null hypothesis. But if your value had been more, say it was 16.50, now it would be somewhere here. So of course then we are realizing that it will go towards the right side. So if it is to the right side, then you reject the null hypothesis. That is a simple way to remember it, that if your value is more than the critical value, then you reject the null hypothesis. And what is the null hypothesis? The null hypothesis in this situation is very simple. It is there is no significant difference between the observed and the expected values. There is no significant difference between the observed and the expected values. Please remember that, and we'll continue with this in the next video. Thank you.